Okay, thank you very much. Can everyone hear me? Is this microphone working? Okay, wonderful. Okay, perfect. Yeah, so I'm going to be talking about compositional representations for flexible behavior. And it's going to be, I guess, a talk which will hopefully convey some intuition, and we'll go to a little bit in the details, but we're never going to go in the murky waters of high details. So that means that please ask any questions during the talk if you would like clarification or something's not making sense. It's totally brilliant if you ask questions. Okay, so what we're interested in is about how to build models of the world. And because we're interested in that, because models of the world really help us with flexible behavior. So here's an example of a rat which has learned that if it goes around this wall, it will come along and find some reward. However, if this world changes a little bit in that the new shortcut appears, repeating these old actions is no longer optimal. And so if I'm just a learner that all I know is to do is to repeat actions, I will not be able to respond optimally when things change. And clearly, the best thing to do is take the shortcut. But that is only possible if I have a model of the world, i.e. if I know that this location relates with that location, that they're really linked. And of course, I'm not just interested in being able to build models of the world for a single environment or for a small change of a single environment. What I really want to do is build models of the world that can map to any environment. And so the real question is, how can I build a model of a new world as fast as possible? Because that's going to allow me to behave optimally as fast as possible. And so one natural way of thinking about this is to realize that each of these worlds can be decomposed into building blocks. So both of them are both in space, so we can decompose it into a building block of space. Both of them have rewards, so we can have a building block of reward. And both environments have walls, so we can have a building block of walls. And decomposing into this way is particularly beneficial because just like how a child can take blocks and make them into a different tower, they can also take those same blocks and build them in a different way into a novel tower. And so that's the sort of the sort of thesis behind this work: is how do we decompose into building blocks and then recompose later to understand any task? And so clearly, the crucial question that we're going to want to address is, how on earth are these building blocks actually represented in neurons? So it's unknown how to do that, but there are some insights, however, from recordings in hippocampus and entorhinal cortex. So for those that aren't familiar, I'm just going to show you what's going on. So at the top, it's going to be things in entorhinal cortex, and at the bottom in hippocampus. And so what you're seeing here is a top-down view of a box. And if this animal runs around in this black trajectory, it's following the black trajectory, and every time you see a red dot, that is when a particular cell fired. So it's when this cell, the cell that we're looking at, is fired. So we can see that this cell is always active, is only active, on this triangular grid, and we can convert that into a rate map, and that's what's known as a grid cell. And there really are lots and lots and lots of these different grid cells, all in entorhinal cortex. They come with different phases and different spatial frequencies. And they really are code for space. They're the building block for space. They tell you where you are in space. You also find other cell representations in entorhinal cortex as well. For example, these object vector cells. These are cells that always respond to, at certain vectorial distances to objects. So here's a cell here that always responds when you're 5 steps to the northwest of all of these white objects. And here's a cell that always responds when you are 4 steps to the west of that object. And again, you have lots of these cells that form maps around objects. These are the building blocks of objects. And it's a similar story for borders as well, cells that form these vectors to borders. And that makes sense, of course, because environments come in compositions, and walls can be anywhere, and objects can be anywhere. It's a compositional situation. You have these compositional representations. And down in the hippocampus, I thought I'd mention you find other sorts of cells, for example, these place cells. These are like grid cells in that they care about a particular location in space, but it cares about generally just one location in space. And you also find these cells like landmark cells, which are like object vector cells in that they have a vectorial relationship to an object, but they don't care about objects in general. They care about particular objects. So I've told you this nice, clean story of these spatial building blocks, but of course biology isn't always so kind to us. And in some situations, you don't see this beautiful factorization into different neural populations for different building blocks. Sometimes you get a bit of a smush. And so what we have here is, well, I apologize, this is meant to be a grid cell. And so this is the first entry into this environment. Oh dear, this is dying. On the first entry into this environment, this grid cell, this is the grid cell. But after a while that this animal's in this environment, it's realized that those 5 black dots are rewards, and it keeps running to those rewards over and over again. After a while, this grid cell field, I don't know if you can see, you can barely see the, I'm sorry. After a while, this grid field has changed from this location. After a while, it's warped towards the reward. And this is interesting because it's no longer a thing that cares purely about space now. These cells are things which care about both space and rewards. We've got this weird smushing of what this cell cares about. To make things more complicated, these very same brain areas that's all about space and spatial understanding has these spatial cells. They're completely critical for when you want to make these shortcuts in non-spatial situations. For example, if I tell you that Jane is stronger than Bob, and then separately I tell you that Bob is stronger than Alice, and separately that Alice is stronger than Gina, and separately I tell you that Gina is stronger than Fred. If I came along and asked you who is stronger, Gina or Bob, you would be able to tell me, even though you've never seen this pair before, you would be able to tell me that the answer is Gina. And that's because you can chain together these pairwise things that I've shown you. You can chain it together and make that inference. And that's just like a shortcut that you've taken that you've never seen before. And it really is the case that, this ability to chain together events is critically reliant on the hippocampal formation. So for example, if you lesion entorhinal cortex, which is the major input into hippocampus, or the fornix, which is the major output of hippocampus, these animals are still able to make those pairwise things that you've told them before, but they are unable to make this chaining inference before. And it's similarly, if you now make, you also find cells in these brain areas that seem to code for non-spatial tasks in a bit of a similar way to how they code for spatial tasks. So for example, if I ask a rodent to hold a lever until a certain sound frequency appears, that's got nothing to do with space. Nevertheless, in hippocampus, you find single cells which care about locations in sound frequency space. And similarly, in entorhinal cortex, you get these multi-peak cells, a bit like a linear grid cell. And perhaps even more compelling, if I show a participant an image of a bird on a screen, well, that bird has a neck length that changes and a leg length that changes. But crucially, it changes as if I was really taking a trajectory around in this bird space spanned by neck length and leg length. But the participants are only ever seeing this bird on the screen. If you put them in a scanner and you show this task, you find this classic hexadirectional signal of grid cells, and that's found in entorhinal cortex and also medial prefrontal cortex. But it seems to be the case that these brain areas, the task isn't in physical space, but it's still coding for them as if it really was in physical space.
Sorry, James, how can you tell those are grid cells for the fMRI?
Oh, so there's, it's a good question, and people have certainly asked that before, and people are never particularly happy with the answer. But there is a particularity. It's a particularity. You're not actually looking for grid cells per se. They're looking for these grid cells conjunctive with head direction cells. And so there are certain directions you can take, of which you'll encounter grid peaks more frequently, and that of course corresponds to a higher bold signal. And if you compare that with the various different directions, you'll end up seeing 6 peaks along the 6 cardinal axes. And so that's what they find. And they find that for if they did a participant walking around a physical virtual reality space and scan them, and they can do exactly the same analysis, and they'll find the same signal whether they're walking around in a virtual world or whether they're seeing this bird move. And so it's a reproducible result. But I certainly agree that every reviewer I've come across has questioned it. Yeah. Nevertheless, this sort of brings about a few questions. Okay. The first one is, how can we have this same system, this hippocampal entorhinal system, that is able to solve both spatial and non-spatial problems and do these shortcuts in space and non-space? Second question is it, we've told this building block story, but is it really the case that these systems are using building blocks to solve these problems? If they are using building blocks, then why do you sometimes get these beautiful single cells that care about a single building block? And why do you sometimes get this smushing situation? And if at all, because we're trying to understand flexible behavior and build models of that, do we learn anything about machines at all? Okay, so the first thing that I'm going to try to convince you of is that it's sequence generalization that unifies understanding space and non-space. And I'm going to show you this in this way. And I'm going to ask you to observe this sequence of observations and actions, so light bulb, right, broom, down, and so on. And at the end of it, I'm going to ask you the banana and you take it up, what should you expect to see? Well, that sounds really quite difficult, but it becomes an awful lot easier if you've seen lots and lots of these same sequences that are drawn from worlds that share the same property, so if there's always a two-dimensional world. Because if you can do that, then you can abstract away these sort of pure building blocks of the space I'm in, you sort of make an abstraction, away from the sensory particularities, the building blocks of stimuli. I can learn that there are these two things that exist, and then they can be recomposed together. Because if I have done this, if I have done this abstraction, then now I can see this sequence, and I can lay it down on this backbone, this spatial building block that I've already learned. And so now when I see the banana and I say, I ask what happens when you go up, I can be able to predict that I'm going to see a light bulb. Okay, so that was for space, and I want to be clear that this is the first time we've ever seen this particular sequence, but nevertheless we were able to make this prediction on first go because we knew this backbone. And so that's true for a world that's structured like space, in which case you need this spatial building block, but it's also true for a sequence if that sequence was instead drawn from something like a family tree. It's not spatial, but nevertheless you can still draw a sequence and the principles remain the same, only I'd have to learn family tree building blocks as opposed to building blocks of space. So that's really the way in which we're going to unify space and non-space, and it's all about understanding sequences as fast as possible, generalizing knowledge about sequences. Yes?
Question. How do you think of sample complexity in the space? You might also imagine that you could just learn the pairwise associations between banana and up.
Yep.
And banana and right. And it might just take you a lot longer to respond adaptively in
Yep.
context. And that wouldn't require like a map per se, but would just require you more exhaustively sampling from the space. Which is to say, non-map-like model with enough samples could give you what looks like map-like behavior, which is just to ask, how do you think of the sample complexity as it relates to model performance?
Okay, so the first thing to say is that the simple case of that associating stimuli with action only works if my stimuli doesn't repeat in different locations, okay? So one, if my stimuli repeats, if my stimuli are alias, I can't do that simple thing. You can still do some fudges about temporal smoothing, but you can't do that simple case. The second thing is that your performance in the case of, if you're wandering around a world and trying to predict things that come up, if you have to learn about every association, your performance will be in line with the number of links there are in the world. However, if you know this backbone, you generalize the link structure, and so your performance will be in line with the number of nodes you see. A number of nodes, at least in almost every graph, is much less than the number of links. Okay, so your performance is much better if you have learned this prior structure as opposed to learning all the pairwise associations. Okay.
But it is the case that every time you give them up from a banana, you choose the light bulb.
No. So I will see many, many different sequences drawn from different worlds. This is the first time I've ever entered into this world. The first time a banana has been followed by a light bulb. But the point was, if I'm learning the pairwise association model, I have to see that link first. Whereas I'm doing the generalization, I don't actually have to see this link. I already know what the consequence of that link is going to be because I know what the structure is. I know if I would be back at the light bulb. Where in the pairwise association, it knows nothing about the structure. It's just associations.
So it's just the sequence of arrows here that tells you.
Exactly. It's the way these arrows add up with each other. And the ways these arrows add up is going to be different depending on different structures. But yeah, we're in a simple case. It's essentially just a sequence of arrows. Yeah, and that tells you where you are. Okay. So the question is, can we build these ideas into a model? And I'm going to try to convince you that, or try to show you that we can. Okay, so we're going to be, again, looking at this sort of like sequence problem. And we're going to be trying to build a model that has this separation between these ideas of building blocks of the structure versus the building blocks of stimuli. And we're going to see that we really need these sort of additional two insights. We just need these additional two things to solve this problem. The first one is we really need to, if we're understanding this abstraction of a family tree, to be able to make predictions, we really need to know where we are in that family tree. We need to know where we are in that abstract tree. And so we need some sort of representation of where we are. And this representation of where we are, just as said, we need to obey these rules of how these arrows add up. So when I see a relationship, maybe sibling, I need to change my representation of where I am to hopefully move on to over here, for example. And when I see the representation of niece and nephew, I need to change my representation to match up with over here. And crucially, when I now see the representation of parent, I need to have the same representation that I had initially. This is called path integration. How do I integrate my actions to know where I am in the world? It's just path integration. And that's been studied for a long time, and you can just solve that sort of system by a recurrent neural network of the sort of equation you see here. Where, of course, this W and B, these recurrent weights, and the way you're adding velocity will be different depending on which building block you're path integrating. And that's just one part of the story. That's all in the abstract space. But we need to connect this abstraction to reality. And so we also need these fast associative memories. Remember, this is the first time we've ever actually seen this particular sequence. And so we need to bind a memory of what we're seeing to this abstract location. And we need to do it for the first step, the second step, and the third step. And now when we're asked what happens when we at the ear and we go right, we're going to path integrate this abstraction. And so now to make the right prediction, we need to come along and retrieve this memory that we made earlier. And so again, two components. We just need something to tell us where we are in this abstraction, and we also need these associative memories as well. Okay, but this is, again, how do we actually go about learning this? And I'll fill in a tiny bit more detail here. So again, we have this model, we have this path integrating RNN, it's all about understanding abstractions. And then we also have these building blocks of stimuli, and they get smushed together, they get put together, in these associative memories. And we're going to do that in a Hopfield network. And that's where now cells in this Hopfield network care about both where I am and also what am I seeing. So each cell is a conjunction, it knows these two things. And then we can come along and we can store memories between these synapses. So like Hebbian learning, we increase synapses between coactive neurons. Okay, and so again, once we've done that, we can do that for memories at every step, and we can carry on, we can path integrate our merry way. And again, just to remind us, these are the weights that we really want to learn, these path integration weights. And then we can ask, we want to make a prediction. So we can predict, and we come along, we have the same representation as before, so we try to retrieve the same memory as before, and we make a prediction of a light bulb. So in reality, it turns out this light bulb was right. We got the prediction good, that's good for us. But of course, if the prediction was wrong, if we got it wrong, that's now an error, and that's an error that can drive our learning. And we can just backpropagate that error through the whole network, it's all differentiable, and we can learn. And we'll learn, that error will help us make these W and B weights better for the next time. Okay, so that's what we're going to do. Now the question is, does this actually work? So if it worked, just as we were saying before, our performance, if we enter this world, in this hexagonal world, and we take this tortuous red path all the way around, by the end of it, we've taken that path, we should know everything. It's in line that our performance should be in line with the number of nodes that we've seen, not the number of edges, because we've hopefully generalized that notion of these edges already. We've transferred that from one sequence to the next. And so that's exactly what we see. This model, the red line is this perfect model which generalizes on nodes, and the blue line is the one which understands edges. And so we can see that this model is in line with this, in line with a number of nodes. Okay, it's not quite perfect, but it's not far off. Okay, so we really are generalizing close to the best we could be. And so that's in space, but it's also true in these family tree structures. It works as well. Again, you have to learn a different building block.
Understand the last graph?
Yeah.
Yeah.
Exactly. So we're now observing the x-axis here, we're observing a sequence of length 3000, so I should make this clear. And the red dashed line is walking around in this A graph. It will be bigger than this one. And the red line is what proportion of nodes we've visited of that whole graph. And the blue line is what proportion of edges we've visited in that whole graph. And the grey line is how good we are at making predictions. And so you can see that our predictions are in line with the number of nodes we have actually visited. I don't know if that makes more sense.
Okay. But the number of total visitations is a lot more than the number of nodes. I guess that's what I'm...
The number of total, what do you mean by... yeah, exactly.
You know, I mean, I'm looking at this, there's like, I don't know.
So yeah, the wall will be bigger. It'll have less than over the order of 150 nodes or something like that. But yeah, I agree. So in this case, it only takes 1,000 steps to visit every node, but it takes nearly 2,000 steps to visit every edge. Yeah. But the point is, after we've done those, we visit every node, we're already close to perfect prediction, okay? We have not done this pairwise association thing. We've really generalized this structure. You can only be this good node predictor if you've generalized structure, and we've done that.
So, but just one thing I'd just like to be clear about have you seen many cases and then you're just generalizing to
Yeah,
new
absolutely.
nodes?
So we've seen many different environments, and so that's going to be again they're much bigger than this, but they have just same backbone, but the sensory observations will all be permutated, and we've just taken sequences from those ones, and so we've seen many different sequences, but we've never seen that environment before. It's a totally novel environment. We've seen sequences that are drawn from the same back. Yeah.
Thank you.
Okay. Okay, and to get to your point here, this is now a sort of a learning course. So right at the very beginning here, this blue line is when we've only ever seen one sequence, okay? And so we're really, really rubbish at generalizing knowledge because we've only seen one sequence, so expectantly it's not going to work on another sequence. But now we've seen lots and lots of different sequences drawn from different environments, and we've been able to learn that backbone, that building block, and now we're essentially close to perfect prediction, okay? It's sort of learning to learn, a meta-learning plot. Okay, it's all very well, but so it works. But the question is, what do these representations? What representations has it actually learned? Okay, and we're going to look in this path integrator, and we're going to see that it learns. And so the top should be model will be what the model has learned, and the bottom is real data. So it learns things that look a bit like grid cells, and they come in different frequencies. They come in different phases, like real grid cells, and then we also learn things like band cells. Okay, and so this really, this really is, again, these sort of building blocks of space. It's learned the building blocks of space. Now, if we put it in environments where there's lots of different objects in different locations, these objects are compositional. Now the task is lots of objects in different places. Then we also learn the building blocks of objects. We learn these object vector representations. So here's one that cares about being 5 north, and here's one that cares about being 4 east. And again, that's very similar to these object vector codes. We've learned the building blocks of objects as well. And the same thing for borders. If we have lots of borders, we learn the building blocks of borders. And again, we need to do this because environments really are consist of objects in different places, borders in different places, etc. So we want to learn this compositional representation. An object, it doesn't look any different. It just has a special signal saying you're an object, and in fact, actions are biased towards that object. So there's a benefit to learn about this object because you're more often gonna go there. Okay, and so that was up in this path integrator. What happens down in hippocampus in our memories? So we also find things, again, model cells, real cells. We find things that look like place cells, so little place cells, big place cells. And again, in the environment with objects, we find things that look a bit like landmark cells. So things that have a vectorial distance to an object, but not to every object. Okay, so what I've just shown you there is representations of just in a single environment, but this is a model that generalizes, so we're perhaps interested in how these representations generalize. So again, looking at this RNN, so here's a grid cell in one world, and the same cell, it's still like a grid cell in the other one, and the same for this other cell. So grid cells, they generalize their shape. And in fact, they just shift and rotate, and that's exactly, I don't have a picture of real data, but that's exactly like real data. Within a module, grid cells, they just shift and rotate. And so that's like saying that 2 grid cells that fire next to each other in one world, they still fire next to each other in another world. And it's the same story for these object vector cells as well. An object vector cell in one world has the same vectorial relationship to an object in the other world. Okay, and the same.
Are the World 1 and World 4 having the same backbone?
They have, yeah, same, but we're still in space. Still, we're just, everything at the moment is in space. Yeah. Okay, and so we can think about what happened.
The same structure in space, they just have different objects.
Yeah, same structure, different, like same like box, but different colors and yeah, etc. Yeah, different objects, or different permutations of objects. Yeah.
The particular paths are
Paths are all,
going all over the place.
yeah, all going. Paths are random.
Just
Yeah.
doing this.
Yeah. Paths are random. Yeah. Okay, so and how do these hippocampal cells remap? Okay, so again, and I haven't actually shown you real data, but again, model cells, a place cell that's up here, then moves over here, and a place cell over here that moves over here, and it really does look quite random, and that's actually also true for data as well. And so 2 cells that fire next to each other in one world do not necessarily fire next to each other in another world. So it looks, as I said, looks random, and at least a large view in the hippocampal literature is that it really is a random place cell map for every environment. but we can question if is it really, because that random in every environment, it does not conform with this building block story. This building block story says these hippocampal cells, remember they combine information about where I am, the building blocks of space and the building blocks of stimuli. And across different worlds of stimuli, and it's just a different composition of stimuli and building block. So that makes the prediction then that the hippocampal cells, after you've remapped, after in a new world, they should remap in a consistent way to what their building block inputs are telling them. So they shouldn't actually be random. They're sort of remapped, they're controlled by the building blocks. I'm going to spell that out in the sort of most extreme way. And so now if I have a hippocampal cell that remaps over here, we could say that actually it's being controlled by this grid cell. This grid cell says you can be active there. And so when it remaps, this hippocampal cell can remap, but it can only remap to another location consistent with this grid cell, which is saying you should only be there. We can then look for that signature. And we're going to look for the signature of what is how, for every grid cell place cell pair that we, in a simultaneous recording of grid cells and place cells, it should say there's a correlation between what is the grid cell firing at this location of place cell peak in one environment with the grid cell firing at the position of place cell peak in the other environment. And we can look for that. And we're going to do that in two separate data sets. As I said, data set of simultaneously recorded place cells and grid cells. This is one is in a physical world with animals just moving around. And the bottom one is actually in a virtual reality world. And we observe this correlation, and these correlations survive a pretty hefty permutation test. So it really does look like the case is that these place cells are not actually remapping randomly. They really are controlled by something analogous like these building blocks. Okay, great.
Sorry, that's a pretty low correlation. Are you saying some of them aren't remapped?
So if you remember, what this analysis is done with every single grid cell place cell pair. Okay, and so that it may not be the case. It might be the case that place cell is only controlled by one grid cell, and so that will, being the fact that we're looking at every place, because we don't know which ones are actually connected. Being the fact we're looking at every single pair will always make the correlation look lower. Okay, but yeah. So that was mostly just things in space. We're just wandering around to objects. You're not doing anything exciting or interesting, but we can make things a little bit more exciting. We can have situations where we're in space, but we're doing a non-spatial task. And so here's an example of that. And so we're in an animal, it's a spatial alternation task. So we're in a spatial maze. It's property of space, but we're going to have a non-spatial behavior. In fact, so we're going to go in here, we're going to go left, and then we're going to go right, then left, then right, then left, right. We're just going left, right, left, right, left, right. And so the animal's rewarded if it goes left, right, left, right, left, right. And it's not rewarded if it goes left, left. Okay? So it wants to alternate. And it's not very clear from this diagram, but however, what you see in the hippocampus is you really see cells. This is one cell, and we're looking at just this central section here. One cell, it only fires this one only fires if you're going left, and it doesn't fire if you're going right. Whereas this cell does not fire if you're going left, and it does fire when you're going right. It is separated space. It's not a place cell anymore. It splits whether I'm going left or right. It's true that they also do find place cells, but they find these splitter cells as well. And this model does exactly the same thing. It learns space because we're in space, so we need the building blocks of space. Remember, these are hippocampus, so it's sort of the conjunction. It's derived from these building blocks. But we also learn these splitter-like cells in hippocampus, and that's derived from these building blocks of task as well. And we see that in a variety of more fancy spatial alternation tasks. So this one, you don't just go left and right. You go out, then in, then out, then in, and in. Okay, you do the whole sort of thing. But the thing's still true. In both this model and also data, you see things that care about space, and you also see things that care about tasks, building blocks of space, building blocks of task, and again, in a more fancy alternation. It's also not just alternation tasks. You can have other sorts of non-spatial behaviors in space. So for example, again, we're in a loop. We're going around and round and round. So we're in space, but we're only being rewarded every 4 laps. We go lap, 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 reward, lap, 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 reward, and so on. And so there's something spatial, we're in space, and there's something non-spatial, we get reward every 4 laps. And so what is found in hippocampus is that you find cells, so this is like lap 1, lap 2, lap 3, lap 4. You find cells which respond the same in every lap. That's a place cell. It spends the same in every spatial location. And you find cells which respond differently in every lap. So this cell is a lap 2 cell. This cell sort of ramps its activity. And this is exactly what this model finds as well. Building blocks of space, building blocks of tasks, and again, in another more complicated setting as well. Okay, so hopefully we've seen a model, and it learns lots of sort of representation. It binds together space and non-space, and it learns lots of representations that one finds in the hippocampal formation and entorhinal cortex. But I just want to take a little step back and think about what we've done from a slightly higher level. And in fact, yeah. So what we've done is we built this building block model, and it learns within a single sequence. Okay, we take a sequence we've never seen before, and we learn something about that sequence so we can make predictions. Okay, never seen the sequence, but I've learned that really there should be a bed here, and so I should predict it later. I'm learning within my sequence, but it's able to do that learning, and it makes that learning becomes better because it has learned something across many, many, many sequences. It's learned that backbone across many sequences, and that helps it learn that backbone helps it behave optimally for a new sequence. This is meta-learning. We've learned the commonality of something, so we can do some better learning within the sequence. Yeah, this is meta-learning, and of course, things like transformer neural networks are all the rage at the moment, and they're, for example, they're particularly good at meta-learning. People in the transformer literature call it in-context learning, but really it's just another type of meta-learning. What I want to now show you next is that this building block model that we made is really actually just a transformer in disguise. Okay. And so for those that aren't familiar with transformers, I'm just going to do a small recap on it. And I'm not going to recap the whole thing, but I'm just going to recap this process called self-attention. And so transformers are a sequence model. And that they take a sequence, it's generally, well, they were originally developed in language, and you're asked to predict something. And what they do is they take each input, and they form a query, key, and a value, a linear transformation from this representation. And they do that for every item in this sequence. And now when I come along and I want to make a prediction, I don't know what this is, but I make a query, and then I just look back in time, and I compare this query to all the previous keys. So I look back in time, I say, oh, well, I don't know what's going on, but I'm going to look at everything else. This is an equation. Look at everything else, do an inner product, turn that inner product into a probability distribution over time steps, and then I'm going to update my own value based on how much I care about each sequence. So I'm going to take this value and say how big a probability have I assigned that, and then just update my prediction. This process is called self-attention. I have a whole sequence, and I'm sort of attending to myself. I'm attending to bits in the sequence. And then this can go through some ML, some nonlinear, some other sort of neural network, and you can stack these sort of self-attention blocks on top of each other. But we're just going to consider this thing right here. And one thing I haven't mentioned now is that, of course, sentences are ordered, and so we want to have some knowledge about what order, which word came first. And so what they do in the literature is they just add a positional encoding. And they say, no, this is number 1, or number 0, this one's number 1, this one's number 2, this one's number 3, and so on. They just say where you are in this sequence. And that's at least as much as I want to tell you about transformers. And so now I'm going to try to show you there's some relationship between the model we've got and transformers, and it might not be totally clear because we've got this path integrating recurrent network, and then we've just got this Hopfield network. But it is the case, however, and I apologize for maths, but I thought this was maybe the best way of doing it. It is the case that if you try to translate and boil this whole model down into its nuts and bolts, its sort of core elements, you can discover that the predictions that it makes follow this equation. And that makes sense, right? Because I'll try to explain it. Because if I want to make a prediction, my query is my next belief of where I think I'll be. Oops, sorry. That's my query here, this G, that corresponds to that Q. And then I'm going to look back in time at all the other locations that I've been to in the past. They're going to be in this matrix G. And then I'm going to compute how similar my query is to previous locations I've been. And then I'm going to make my prediction based on how similar is my believed location to locations in the past. And I'll just retrieve the thing, the observation that was bound to that. Okay, so we can see there's some sort of thing in spirit between these two equations. In fact, this is just this equation minus this softmax, and where the query is my belief of where I am, and my values are the observation. And so we can make some analogies now. And it turns out these Hopfield networks really are just the same thing as the self-attention. I should say I haven't got the citation here, but that is also that was definitely not me that did that. That was known. That was shown at roughly the same time, so we know this Hopfield network, this memory thing, is really performing the self-attention. And what corresponds to the position encodings is this belief of where I am. This belief of where I am is a position, right? In our case, it was space, where I am in space. But in this case, it was like 1, 2, 3, 4. And so it's really the case that this position encoding is like the grammar. It's like grammar of where I am in this sort of this sentence, where I am in this argument that would correspond to this. So perhaps make some statements about the position encodings that given in transformers maybe are not the best ones. But of course we've missed one thing out. To make this transformer really into a model analogous to our model, we'd actually need to add these recurrence between these position encodings. So we need to have these recurrence between the position encodings, like we've got the recurrence up here. Okay, and so we can sort of play with that a little bit, and we can think, right, well maybe we can actually just replace our hippocampal model over here with a transformer that just has these recurrent positional encodings. Okay, and so we're just going to do that and see what happens. The first thing to note is that including this softmax term massively improves learning. Okay, so here's the case of our the situation we had before, and it sort of slowly learns, and it will continue on getting better and better and better, but it takes a long time. But the situation where we have this softmax, it just learns super fast, and that makes sense again, because as soon as you turn things into a probability distribution, it becomes an awful lot easier to attend to things in the past. Whereas in this case, we'd have to also learn some nice normalizations and make things all orthogonal. But the softmax sort of alleviates that problem. The second thing to notice is now we've got this transformer, just these recurrent position encodings. It also, and we train it on the same task as before, it also learns spatial representations. So we learn representations that look like grid cells here, and here are the autocorrelations of these cells. And we also, again, learn things like band cells. I didn't train it on the situation with objects, so we don't get to see them here. But we do, nevertheless, it still learns these building blocks of space in that position encoding, in the grammar. It's learned the grammar of the situation. Okay, so this next bit is going to get slightly more technical, and so I apologize, but please ask questions. But what we've discovered here is we've learnt the structure, we've learnt the backbone. The question is, do we have an equivalent to the hippocampal place cells? Does that exist in this model as well? And it's a little bit harder where it comes from, but it does actually come from implementing this softmax. So when we had this softmax, remember this replaced our hippocampus. We didn't need a hippocampus anymore when we utilized this. But it turns out if you actually try to implement this softmax neurally, you'll end up with things that look a bit like place cells. And I'll try to explain that now. We've got a query vector here. This is our query vector of what we're seeing. And then we can implement this query multiplied by our lookup when we're looking at back in time. We can now implement this in weights in a network, where this can be with the first memory we stored, this is the second memory we stored, and the third memory we stored, and it each projects to its own neuron. And so that's and then after we've made that projection to each neuron, then we can do this softmax. Okay. And of course, after we've done that softmax, then we likewise will connect back to make our prediction, and that's just again where we have memories x1, x2, x3 corresponding to then they're linking up to the right memory neurons up here. Again, so once we've implemented the softmax neurally, or in a possible neural way, then we can come and think about then what are the representations, what are the rate maps of these memory neurons, and they then start looking a bit like place cells, and that makes sense because we only want to retrieve memory 1 in a certain location, and we don't want to retrieve memory 1 over here, but we want to retrieve memory 2 over here. And so again, we get these sort of place-style responses in this memory neurons. And this is really a bit like hippocampal indexing theory. Hippocampus is indexing these cortical memories. And again, when we move to a new environment, these cells remap, and again, they sort of remap in in whatever way you want, really. So, but what's interesting is it does not remap in that. Remember, I showed you that prediction earlier. And those conjunctive cells, they remap consistent to the building blocks. This does not do that. And so perhaps it's some evidence that the hippocampus is not a transformer. Yeah, please.
I think I've lost track of what the memory neurons are supposed to be.
The memory neurons are just linking, they're just like effectively serving the role as an index to cortical memory, to cortex. So this is a cortical representation and another cortical representation. It's like at a point in time, this G1 was a memory at time step 1 and of this representation, and X
Buffer for whatever activity was happening.
Exactly, it's just a buffer. Yeah, exactly right. It's just a buffer. Yeah, but of course you don't have to, because some of those memories will be the same when I return to a location. You don't have to update that buffer, for example, every time, every step.
The memory is just of the activity in whatever.
It's just a memory that binds together the current activity at the current time step. Yeah, exactly right. Oakley Dokes. Yeah, so I just want to say this has a very different interpretation to place cells, and so it perhaps, being that it doesn't agree with data, it perhaps implies that this interpretation is not actually right. But nevertheless, it's perhaps an interesting analogy to make, and it also makes reasoning over some other things a little bit easier. For example, how do we how we bind together many, many different building blocks. We don't just bind together space and where am I and what am I seeing. We can bind together lots of other things. I'm just going to, for the purpose of this, I've labeled this X, but for the purpose of this next schematic, let's assume that X is actually really an action. I'm binding together what action am I going to take at each location. That's just for this particular schematic. But again, as I said, we can now bind together multiple different things. So for example, I might want to bind together a context. Like in one situation, I want to do one thing, and another situation, I want to do another thing. So this can now be a context signal that I bind together. And that's like saying that now I don't just saw G1 with X1, I saw G1 with X1 with the current context. And maybe my context is in this situation, I always go right. But of course, I can make another context and make new memories, and in these other locations, 1, 2, 3, 4, I always go down. I always take this second action, that second action down. And I can just arbitrate between them, between this context signal as well. And that's really easy to implement. You just add on a context, which now attends to previous context as well as previous spatial locations. And I can remember that I always go right in one location, always go down. So always go right in one context and always go down in another context. And so,
sorry, I'm just trying to understand this like intuitively from
yeah.
a biological point of view. Like that this means that a single neuron is going to respond to multiple stimulus, like become more conjunctive.
No, so remember all of these, so each one of these things here, each one of these bindings will just correspond to a single one of these memory neurons. Okay, so this memory corresponds to this binding of G1, X1, and T1, and this one will correspond to another binding. Okay, so you just, in this case, you just need 8 of these neurons, and they do the 8 different bindings. Okay, sorry if I've lost you, but the last slide on this, and that is to say that this is quite analogous to top-down attention, and it relates to things like the Miller-Cohen model, which is a model of how prefrontal cortex controls or gates representations in lower cortical areas and reroutes information depending on context, for example. And so in that model, you sort of view it as train tracks, so train tracks of information flow, and they get changed, the direction of those train tracks get changed depending on a context. And that's exactly what's going on here, only we're doing top-down attention over memories instead of other sorts of representations.
But you don't need to change anything about your model here, the transformer architecture.
No, you don't have to change anything. The only thing you need to change is you need to just add in this term. Everything else is the same, yeah. But again, it just makes this idea, these are all just different building blocks. And so all it says is like, for this building block thing, it just says the inputs to your transformer just want to be those building blocks. And that's all that this says. Okay, so we'll recap a little bit what we've done. So we've seen that having this building block idea ends up being quite helpful because we can understand new situations as fast as possible. And we've also built this model, which is a bit transformer-like, and it learns these different building blocks in different neurons. So it learns a neuron for space, neurons for objects, neurons for borders. But I sort of cheated you a little bit, and I apologize for that. And that's because when I learned neurons which only cared about space or only cared about objects, it's because that was a little bit hardwired into the network. I said you can only learn about one thing and you can only learn about another thing. I hardwired it. So it doesn't really tell us an awful lot, because actually it would have worked just as well if those representations were a linear rotation with each other. Okay? Building blocks they're all still there if I just rotate that whole population. Okay? So I take a whole population of grid cells, object vector cells, and border cells, and I rotate them. That still has all the same information as building blocks, and it would have worked just as well. So we haven't really learned anything about why do we sometimes see these in the real brain. Why do we sometimes see these pure building block coding cells, these cells that only care for space or only for objects or only for borders? Why do we see that in some situations, and other situations we see this sort of weird warping, this smushing situation? We just haven't answered that because the model was hardwired to learn these separate building blocks. And I just want to highlight this phenomena of some.
Sometimes in some situations you see the pure ones, and in other situations you see the.
So I will hopefully explain it later, but maybe as a preface, a prelude, you generally see this situation where things are very compositional, i.e. the objects occur in many different locations, etc. And this smushing happened to be the case where this animal had just basically it's found these rewards and it just hangs around these rewards all the time. It does loop, 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 loop. It becomes like the task itself becomes intertwined between objects and space, and then the representation becomes intertwined with objects and space. So one situation generally is when you need to be compositional, and the other situation is when you don't need to be compositional.
A mix of these. In fact, it's very rare to see things that happen.
So I agree it's very rare, but it's also so it's the case that if you go into entorhinal cortex and you say you look at a cell and you say which can I decode lots of things from this cell. And sometimes, I agree, you sometimes see these pure cells, but you also see cells where you can decode space and head direction from, for example. But I'm just going to say it's really hard to interpret that, okay? Because there are, of course, if there was a situation, you're only trying to decode the set of variables that you think the animal might be doing. But there are many other variables the animal might be doing. There's some situations where head direction and space are naturally entangled because the animal might be where they urinate and they don't want to head over there very often, for example. So we do not actually know what the variables of interest that the animals are trying to represent are. But nevertheless, it is the case that you do see, I think it's, well, I don't know if it's been shown, but I think you see these cells more common than you would expect. You see these pure cells more common than random. Okay.
It's always it's a continuum. There's no such thing as like such a discrete categorization. People make it sound like that, but it's never like that.
Okay,
if you actually look at the data, it's always a continuum.
I'm happy to buy that, but
Regardless of context.
I'm happy to buy that, but I'm also the case that it's really hard to know what that continuum is because we do not know what the variable, the building blocks of interest to that animal are, and it's also the case. What you'll hopefully see later is I don't mind it being a continuum. I'm happy with it being a continuum, but I just think that these things observe more than chance. That's what I care about. Okay.
So the last, things being more continuous and discrete debate is maybe not as helpful to me as just this question. You said that the task structure changes these two representations. Moves you more towards one side than the other.
Yeah.
Can you just, are you going to review some evidence of that particular fact?
No, the only evidence I have, the only evidence I'm going to say is for this case, and this situation of this grid field warping really does take a long time. It really is after this animal is just, so the task is actually in this animal to go to collect this 1, 2, 3 award and go back to start and then 1, 2, 3. And the animal, all it does is just 1, 2, 3 loop, 1, 2, 3 loop, 1, 2, 3 loop. It does over and over and over and over again. And this warping is slow. It slowly warps over. It's
overfitting very.
It's overfit, exactly. It's overfitting. And you're overfitting, you're learning this, like, you no longer need to learn space because you're learning the space and reward combination.
It's less compositional when you
exactly. Yeah. I don't. Yeah. Exactly. Yeah. I don't mind too much about this discreteness. All I care about is it's less compositional.
Okay.
Yeah. Exactly. Yeah.
So I don't know whether you accept that point or not. But, no. Okay. We'll have to discuss it with you later.
Okay. Well, I hope you accept this grid cell did move. It changed. It changed its representation. Okay. 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 It's something.
This is a discovery that was first made in the hippocampus, and this is just in grid cells. That's new about this. That people thought grid cells didn't change. I mean, this is not particularly surprising to me. If it wasn't like this, it would be very surprising.
I guess I don't mind whether it's surprising or not. I just care that it happens. And it really is the case that this grid cell changes. Its understanding changes from one situation to the other. It is not the same grid cell in every situation. That's all I care about. It is not the same in every situation. It changes depending on the situation.
The question is, like, why do you emphasize this though, when there is so much variability
because
in the campus and everywhere else in the entorhinal cortex, and what makes this very compositional and not anything else?
I emphasize it because the animal's behavior becomes non-compositional and the cell itself changes. That's what I care about. I don't mind that it's, all I care is that this animal behavior is very entangled with the space and this cell changes to become entangled with the reward as well. That's all I mind. I don't mind if things are on a gradient as long as they're less compositional there and more compositional here. Okay, I'm happy with that. Okay.
Map, you can learn it just by sampling exhaustively.
Yeah, you could do it. Yeah. Okay, well, if you didn't like this, I'm sure you're going to hate the next few slides. Okay, but the point is this phenomenon isn't just unique to the hippocampal formation as well. In parietal cortex, for example, you get some cells which only care about one task. So this cell only cares about this T task, and this cell only cares about this SW task. And across the whole population, you have a bunch of cells that only care about one task and a bunch of cells that only care about another task. And it's also true in prefrontal cortex. But then sometimes you also find cells that seem apparently mixed selective in prefrontal cortex. So this idea of sometimes things being pure selective and sometimes things being mixed selective is really endemic across across these three brain areas that I've shown you. Okay. This whole thing is also a bit of a big deal in machine learning, because they are interested in observing data and trying to work out what are the underlying factors of variation. So for example, they often make the assumption that their data is generated from a set of independent random variables, x, y, and z, and they go through some data generative pattern to produce some observations. And crucially, when I say independent here, it's the same as compositional. Compositional just means they can occur in any combination. That's also what independent means. And of course, these machine learners, they don't have access to these data, and so they have access to the observations, and they're trying to infer some underlying latent factors. And they tend to do this with, well, at least in vogue at the moment, is to do this with autoencoders or variational autoencoders, which are these neural modules where they take the input, they project it up into some latent space, and then they put all sorts of constraints on that latent layer in the hope that those constraints are going to make it going to make a single neuron care about a single thing. Because if they can get that situation, then their model becomes more interpretable, and there's a bunch of other properties that are useful as well. And so there's an example of where they've trained on lots of images, where each image is an object, and the object can have a different size, different x, y location, different shape, and so on. So lots of different images. And what I'm actually showing you here is not actually the data set, it's showing what each neuron ends up caring about. And so this neuron here ends up caring just about their y location. This neuron ends up caring just about the x location. And this neuron ends up caring about the size. And you can look down the other neurons, and they also just care about a single factor of variation. So this latent layer has apparently learned something about this data. And it's also the case, as an interesting example, it's also the case that if you train this exact same model now on faces, and you also you train this model on faces, and you also record in infratemporal cortex neurons when they are looking at these faces, there's something that's not far off a one-to-one correspondence between these model neurons and neurons in IT cortex. So it's perhaps implying that these neurons in IT are also doing this some sort of disentanglement. They're sort of they're decomposing into the building blocks. And so how do we take all this sort of this evidence from the cells, this sort of stuff that's going on in machine learning, and how can we try to how can we try to understand what's going on in those cells? And we're going to try to answer it by, in the same way that machine learners do, they put these constraints, and so we're going to try to consider what are the actual constraints on biological neurons, and see if that tells us something about why, in some situations, things should be modular, and in some situations they shouldn't. Okay, so what are the constraints on neurons? Okay, the first thing is these neurons actually have to be useful. They have to actually represent the external variables of interest. And so, for example, if we've got a neural representation and we care about position, then our neural representation should also care about position in some way. And likewise, if it cares about action, it should also, our neural representation should also care about action in some way. And not only that, but if we, for different positions, we should have different neural representations, and for different actions, we should have different neural representations as well. Okay, so our representation actually needs to represent the things in the outside world in some way. But they also need to obey biology. We're in biological neurons. And so, for example, neural activity can't be negative. That's not good, but this is okay. And neurons, we don't have infinite resources. We can't fire at infinity, etc. So we want the activity to be as small as possible. And we also want, for example, weight activity to be as small as possible. I'm going to try and show you now that these intuitions, these constraints here, actually lead to a neural population preferring to have single cells that care about single factors of variation. Again, I'm not saying it's for sure, it's just saying it's preferred. Okay, so here's some intuition. Okay, so we've got these two neurons, and they're trying to represent these two random variables, E1 and E2. And it's doing it by linear. In fact, our neurons are currently, all they're doing is rotated these random variables. Okay, but they are representing them, that's fine. But we can see at the moment things are negative, so that's not good. Okay, so let's try what I'm saying here. We can see that the neurons are representing these random variables, one's along this axis and the other one's along this axis here. Okay, and we're centered around 0. And this is the energy of our population, so it's low at the moment. But it's negative, and that's a problem. We can't do that. Okay, so we have to be positive, so we can shift everything up. So we've shifted everything up, but that introduces this extra energy. It's made us less efficient in terms of energy. And it's sort of clear that to make this, to represent this distribution, still represent this distribution, but in the most energy efficient way, we want to squish it into the axes as tight as possible. And squishing it in, we can see that reduces this. But what the squishing in means is that each random variable now is really only along one neural axis. Okay, a neuron cares about a single factor of variation. One neuron cares about one factor of variation. As said, that is well, at least from this argument, that's the preferred case. And just to be an arse, I'm going to show you that there is some maths behind this that proves this in well, not all situations, but some situations, enough situations.
How sensitive is this to the kinds of noise you have? You're assuming, for example, random variables.
Yeah. So the assumption at the the assumption is that you have random variables that have a finite support, so we can't go to infinity. Finite support. And that they just, they have a maximum value and a minimum value. So that's one assumption. And there's another assumption. So it doesn't have to be this like uniform distribution. That's not necessary. And the other assumption, at the moment, but hopefully I'll change it soon, is that your the input, it needs to be whitened. That's the other assumption at the moment. Yeah. Yes.
It's like kind of a weird argument to make because it's assuming that there's some kind of truth about what the actual directions are.
What do you mean by?
What the fundamental variables are that the neurons are supposed to be representing. So if you imagine that the fundamental variables were different from what were not space and objects, but were actually some mixture of those.
Yeah.
To satisfy the biological constraints.
That's fine. These fundamental variables do not have to be space and objects. Okay. And they just need to be. This is just saying if you can express your data in terms of independent variables, then your neurons will align to those independent variables. It might be the case that you can set up a situation in space and objects where the independent variables aren't space and objects, but there's some mixture of space and objects, in which case this is saying you would find that mixture. Okay. Yeah.
Okay, but given that fact, it doesn't seem like this result would explain why we see neurons aligning to the particular.
Well, I mean, hopefully I'll convince you that it will, because in the situation where space and objects really are two independent variables, i.e. objects can be anywhere in space, those are the two independent variables. And so this is saying that you will want to have a neural representation that has separate neural populations for each of those independent variables. That's all it says. Yeah. Okay.
So, maybe it's helpful to note that when an animal is exploring an environment because someone sprinkling chocolate sprinkles around them, that's sort of like an IID relationship between, or independent relationship between reward and space.
Yeah.
Right? And then when the reward is always in one place, that changes that. So I'm following that part of the argument. But but there is a feeling that I guess you have that in the real world there's a lot more of these kind of independences than I'm necessarily sure I really think is true.
I don't think everything in the world is totally independent, but I think it is independent enough for it to be beneficial to partition things into building blocks. Because if you don't partition things into building blocks, you sort of have to learn bespoke-ish situations. And if I have to encounter 100 things, that isn't the full distribution, but it's enough that I don't want to have to learn 100 different bespoke things. I want to make it into building blocks. And this says when I make it into building blocks, it's beneficial from an energy argument to partition those building blocks into different neural representations. So, I mean, as an example, I don't know if Laura Driscoll's here, but from Laura Driscoll from Stanford, for example, she can train an RNN on loads and loads and loads of different tasks, and then when she puts a ReLU on that RNN, she'll find subpopulations in that RNN that just care about one task, and they get composed. It seems to be like a pervasive phenomena, at least also in, and I'll hopefully show you some simulations later that it shows that this does seem to make, does seem to get separate neural populations for each variable. Yes. Mm-hmm.
But it's definitely not like probabilistic. Like it's not like language is a bag of words model, where like the covariance of 3 words can just be factored into the independent probabilities for each word individually or in a sentences. So, is there some other notion of independence that we need besides probabilistic independence to capture this?
I don't know. Again, I don't mind if things aren't totally independent. I really don't mind. It's just as long as this idea of separating the building blocks is profitable enough in most situations, then I think it'll probably happen. I know that's not the proof that I've given here, but that my suspicion is that will happen. Like it's impossible from enough data, it's impossible to ever say that you've got independence because your data is always, samples, right? So you can only go to a certain level of confidence of things being independent. Yeah.
So James, this is really interesting and provocative and getting lots of discussion. I want to make sure you.
Yes, I've got. Oh,
chance
sorry.
to finish up.
Yeah, I've probably got about 5-10 minutes left. I don't know if that's okay.
10 more minutes. would be-
Okay, great. Okay, so we're just going to now see, this is an intuition and a proof that was convincing to me, but maybe, yeah, okay. Okay, so we're now going to see if it holds out if we do some simulations. Okay, so we're just going to now simulate some neural networks where we're going to add these constraints. We're going to add these constraints as regularizers, okay? So for example, we might add a constraint that says, don't be negative, okay? So we're going to add a constraint which says, I want you to have as small of activity as possible. I want you to not have really high weights. Again, energy efficiencies. And then I'm going to give it this network a bunch of data, and the data is going to be, our input data is going to be a mixture of random variables, and our output data is another mixture of those random variables. So there really is a true mapping from input to output. But both input and output are mixtures of random variables. And we're going to train a network such that we can make a prediction, and we're going to try to minimize the loss. Okay, and so these are all our constraints that we're going to try to satisfy. And we're going to train this network, and so we've just got a nice linear, at the moment it's just a linear network. And we can see that when we train this network without any of our constraints, I haven't shown, but we've learned this task perfectly fine. But what we see is in this middle layer, that's where our constraints are all in this middle layer, I'm sorry to say. In this middle layer, everything's mixed. And so here's an example of we compute the mutual information between a hidden layer neuron and our factors, and we can see that each neuron cares about lots of factors. Okay, things are still very mixed. But when we add these biological constraints, it really is the case that we get this beautiful factorization, this beautiful separation, this beautiful disentanglement, where single hidden unit neurons only care about a single factor of variation. Okay? And that's true if we did it in, I should say, that's only the case, it's not a great plot, but it tends to be only the case of where we have all of these biological constraints. And when we start chopping them out, things get worse. And that's also true in deep networks. This is a deep network. I'm sorry, this is just terrible because I haven't explained what these plots are, but it's a plot of it's a measure of how well disentangled we are. And we can see when we have linear data, and we've got here 5 hidden layers, all of the layers learn to be roughly this disentangled. And where now we're going to make the process nonlinear, so we're going to have a nonlinear network. Sorry, this network was also nonlinear, but now we're going to have a nonlinear network as well. But now our input data, we're going to transform it. We're just going to take exactly the same input data, and we're going to cube it. Okay, so that's an invertible transformation. And so there still is a real map from input to output, but now it's a nonlinear map. And what we observe is these early layers, layer 0 and 1, they stay entangled. They never get disentangled. The middle layer gets a little bit, but the later layers are this disentangling. Okay, and that sort of makes intuitive sense because we've made this nonlinear, and so now we need to. The early layers, what they're doing is they're undoing that nonlinearity, and to undo that nonlinearity, you have to have some entangling. Okay, because we've taken a mixture and we've cubed it, so we have to. The only way we can unmix it is to still have layers which are mixed. Okay. But towards the end, everything gets nice and disentangled. And again, here's just a plot of a late layer. Single neurons care about single factors of variation. In the interest of time, I'm going to skip this, but it's true that also we can do some unsupervised data in linear and nonlinear. Again, we find these beautiful situations. And now we can do this in a, yes?
Is it fair to say that a corollary of what you're showing here, if true, is that in the brain, the default should be for you to find very disentangled representations, meaning a single neuron should fire only
what this is saying is that if your task is compositional, or if it's beneficial to solve your task in a compositional way, you should find these disentangled representations higher than chance. That's like that's the weakest statement I can make. Yeah, but it's still the case that even though my task is compositional, I might need to do a nonlinear transformation to actually predict the outputs. Okay. In which case, I will have some representation that might be this nice disentangled one, but to do the nonlinear stuff, I still have to mix things up. So it's not saying everything is going to be unmixed. There'll still be things that are mixed, but you'll find
deeper in the brain.
It's just more than random. It's always saying more than random. You should find things that have these modules for single things. Okay, all it says. Okay, and likewise in these networks I've shown you, it's not every layer is not disentangled. It's only some of them. Yeah. Yeah, exactly. As you move further and further away from the input. Yeah. or move further and further away from the thing that is crazy mixed and nonlinear mixed. Okay, so it's also true, sorry, just to make sure I don't keep you forever. This also works in these more machine learning data sets, these benchmark data sets. So here's a data set where they feed in images of objects, and those objects can have different shapes, different colors, different floor colors, different wall colors, different viewing angles, different sizes. And I'm not going to take much from this, but our models never learn these disentangled representations, and they actually tend to learn better than other models, and I'll show you that. And again, you find things which aren't as clean as before, but it is learning single model neurons correspond to single ground truth factors. And here's a bit of a, not the best plot, but it seems like these biological constraints seem to fall in this Goldilocks period up here. These are all the ones with biological constraints, and they both have high disentanglement score, and they also predict the data well. Whereas you get these other sort of models of machine, I didn't do an exhaustive search, because I'm more lazy. But these other models in machine learning, they when they do get high disentanglement, they sacrifice on predicting the data. Okay, so we've seen to fall, these biological constraints seem to fall in this Goldilocks period. And here's just an example of the sorts of representations that are learned. So this one neuron has learned the shape, so the size of the object. This one's learned the color. These two haven't learned anything, so we're actually just going downwards here. These ones learned the floor color. This ones haven't learned anything. This one's learned the wall color, the shape, and the viewing angle. Okay, and now we're going to try to explain this cell data now, and we're going to try to explain it with the same principles we've done. So we're going to give it first one. We're going to give it a compositional task. We're going to give it a task where there's things in space, objects in space that you often head towards, objects in space, but those between different tasks, those objects move around. And we're going to have loads and loads and loads and loads of different tasks, but the objects are always in different places. Okay. And we're going to train a representation. I'm going to train this sort of recurrent representation. I'm going to ask it to predict space, and we're going to ask it to predict these two other things. It doesn't really matter whether we're predicting object and actions, but the point is these two are entangled with each other, but they're independent of space, because objects are always moving in space. But you always head towards objects. So the actions are towards objects, but objects in space are unmixed. Are factored from each other. And we're just going to train this with our biological constraints and this functional constraint as well. Okay, so first, if we do this without a ReLU, so without a non-negative constraint, we find these sort of things that look a bit like grid cells, but they're not perfect grid cells. And in this environment, there are no objects. In this environment, there are five objects, four objects, 1, 2, 3, 4. And we can see that this cell sort of looks pretty much the same between the two environments, but there actually are small differences between the two. And it's the same for this other cell. But however, if we train with this ReLU, so we have a non-negative constraint now, our population really falls into these two categories. We either have a grid cells, which don't care about whether there's objects at all, and you have also these other cells which are not active if there are no objects, and they are active if there are objects. So these cells only care about objects. These cells don't care about objects at all. So this is the sort of explanation that I try to give you, that these biological constraints are a pressure for our representations to fall into modules, where one module cares about one module cares about one building block, and another module cares about another building block. And these are going to relate to these grid cells and object vector cells. Okay, so now we're going to do the exact opposite. We're now going to train on some task where we're only going to see one task now, and so the objects are always in the same location, so they're entangled with space. And we're just going to train on that task repeatedly. And we're going to do exactly the same thing as before. And what we observe things that look a little bit like grid cells. We've got all of our biological constraints at this point. And things look a little bit like grid cells, but the eagle-eyed of you might see that the peaks are always sort of warped towards the object. So this peak has always sort of got the same relationship to the objects, and so on. So we've sort of recapitulated this warping phenomena, because we're just repeatedly learning in this one environment. And this is a little bit similar to this warping phenomena that we saw earlier. These grid peaks are warped towards the rewards, like we saw. And again, if we compare it to the situation where we've got a compositional task, where we had these two separate populations, grid cells and object cells. And last plot just says that, again, if we train a bunch of models with different initializations and so on, it's only these ones where rewards and objects are entangled with each other do we find this warping. Okay, so a little summary of what we just saw there. So hopefully we've gained some formal understanding of why biology prefers separate neurons for each building block. And that maybe explains a little bit why when experimentalists over the history of time, they've gone in and recorded, when they just, Hubel and Wiesel, just recording one cell at a time, that one cell only really cared about one thing. And so maybe it's an explanation for why so often So why so often cells recorded care about single independent human interpretable factors of variation. And then we've also seen that we recapitulate these warped and mixed selective cells when tasks stop becoming compositional. Okay, so I'm just going to return to the overall questions we started at the beginning. So we wondered, can we have a system that can solve spatial and non-spatial problems? And we tried to solve that, tried to say that we could with sequences and sequence generalizations, and we built a model about this. Question, is it really using building blocks? Well, the model seems to then come along and learn grid cells. It learns these building blocks. Well, the place cell's not a building block, but it learns all these sort of building block-like representations. And we also made a prediction that, of how place cells remap, and that was consistent with this building block hypothesis. If so, why are these neurons pure selective and sometimes not? Well, hopefully we've, again, offered some explanation during just the last section about cells, when your data is really independent or compositional, you prefer to have single modules for each variable of interest. And lastly, do we understand anything about machines? Well, I mean, jury's out, who knows? But we've at least provided some biological constraints now which maybe make disentanglement a little bit easier. And we've made some relationship between hippocampal models and transformers and what position encodings might look like in transformers. Okay, yeah, that's it. And so just thank you to everyone who has helped along the way and to funders and universities and so on. Thank you, guys.
We have time for 2 questions, and then you can join the dinner and ask questions.
Okay, so just one quick question. So in your example where you either learn the spatial grid or the sibling parent nephew thing, those are both 2-dimensional representations. How does your model work when it's more than 2-dimensional or in arbitrary dimension?
So family tree is not actually 2-dimensional for a reason, that's maybe not so interesting, but it also works on things that are totally, definitely, no argument, totally non-spatial. Yeah, it has no care, it doesn't care in the world about that. All it cares is the actions that you're providing actually make sense in the real world. So the actions you need to provide do need to correspond to a real action trajectories in whatever graph you're on. Yeah.
Yeah, so the first part of your talk reminded me of the SLAM problem in the Odyssey study.
Yep.
Simultaneous localization of matter. And one of the things they care a lot about in that problem is propagating uncertainty about where you are in the environment. And I noticed you don't have any representation of uncertainty in your settings. And I'm just kind of curious if you've explored that and how that might change the types of representations you expect.
Have not explored it at all, and yeah, you raise a good question. So everything is totally deterministic, and so not only the model is deterministic in that we're using an RNN and no noise, but also the environment. Every time I take a right, I definitely go right, for example, and every time I'm at a location, I always see the right observation that should be there. There's no noise whatsoever. I haven't thought about, but I would happily talk to you about it, and maybe you tell me what I should find. And yeah, okay.
Okay, maybe one last question.
Hi, James. I'm Laura.
Ah, hello.
So yeah, it sounds like it's really cool how you show in the end how these biological constraints lead to the particular representation that we see. There's some understanding of like why we see the things that we do. And one thing that you showed throughout the talk is that there's these grid cells and place cells that show up a lot. Particularly with the grid cells, do you feel like by training these networks to do path integration and seeing them define grid cells, does that develop any intuition for why it's this particular kind of representation?
So why does a grid cell look like a grid cell?
Yeah.
So yeah, so I do have another piece of work he's not here. Another piece of work which tries to, well, I mean, me and other people try to delve into why a grid cell looks like a grid cell. So it turns out those 3 constraints that we were looking at before are you need to encode the variable, so you need to encode where you are in some 2D world somehow, and that you need to be non-negative. So Ben Sorcha over there is also a proponent of non-negative with grid cells. But we've got a sort of a mathematical analysis. So if you care about where you are in the world, you care about being non-negative, and you care about having not really high energy efficiency in your representation. We have some sort of mathematical arguments that show that grid cells are actually optimal in that case, in that situation. But the key is that, the key that I missed as well is that this notion of path integration is totally fundamental. There needs to be some representation that not only do you represent location, but you also need to have the ability to move from one representation to another, and move to another, and take you back into a loop. Your representation should go back into a loop when you really go back into a loop in the real world. And so those constraints, you can analyze them and show that hexagonal grid cells are optimal.
Great. So due to the limit of time, that's all the questions we can take. But you can still ask questions over the dinner. So now please join me and thanks Dr. James for the talk.